Welcome to Tech News at FlowConnect YouTube channel. CRISPR can edit human genes in vivo, according to recent research. In the past, this method was primarily used for altering cells in vitro, away from the patient. The potential for more immediate treatment of diseases is being explored in recent research. Biologists Jennifer Doudna and Emmanuelle Charpentier made headlines a decade ago when they described a bacterial immune system's potential as a tool for modifying the genomes of live organisms. In 2013, scientists led by Feng Zhang at the Broad Institute of MIT and Harvard said they have used the CRISPR technology to modify cells from humans and other animals in the lab. Both groups' efforts sparked a renewed focus on using CRISPR to cure genetic illnesses, ultimately earning Doudna and Charpentier the Nobel Prize in Medicine in 2020. Since many diseases result from faulty genes, CRISPR's potential to snip out or replace an aberrant gene holds the promise of curing these disorders. However, getting the gene editing components to the area of the body that needs treatment has been a hurdle in translating CRISPR findings in the lab into treatments for patients. CRISPR Therapeutics, a biotechnology firm, has circumvented this problem by modifying patients' cells in vitro or outside the body. Patients' red blood cells are removed from their bodies, edited to remove a disease-causing mutation, and then reinfused. There, doctors have used the device to treat many sickle cell anemia and beta thalassemia patients. However, there are drawbacks to working ex vivo. It's hard to implement, expensive, and ineffective. Most illnesses reside in parts of the body that are difficult to isolate, treat, and reintroduce. Therefore, the next stage of CRISPR research will concentrate on editing, in vivo, or within the living body of a patient. This was initially shown to be feasible for transthyretin amyloidosis last year, and Intellia Therapeutics was the pioneer in this field. The biotech firm based in Cambridge, Massachusetts, recently demonstrated in-body editing for a second ailment. 6 patients with hereditary angioedema improved their symptoms after receiving the business's CRISPR treatment, the company said at a conference in Germany. In a second statement, the company reported that 12 patients suffering from transthyretin amyloidosis, a hereditary disorder that can cause deadly heart failure, had a reduction in a dangerous protein of more than 90% after receiving one of its CRISPR treatments. These findings expand upon a trial involving 6 individuals published in the New England Journal of Medicine last year. CRISPR successfully and safely modified both genes involved in the illnesses. John Leonard, CEO of Intellia, remarked, "This shows us that we may have the same kind of effects in a completely different gene." Even though the 2 trials were modest and the most recent results have not been published in a peer-reviewed publication, Yan Zhang, an assistant professor of biological chemistry at the University of Michigan who researches CRISPR, calls the findings an important milestone for gene editing. She concludes that, overall, Intellia's recent positive studies offer promise for employing CRISPR therapies to modify genes directly inside the human body. Because the CRISPR components can't enter cells independently, Intellia employs a delivery mechanism called lipid nanoparticles, microscopic fat bubbles, to transport the CRISPR components to the liver. Patients in Intellia's trials get a single intravenous, IV, injection of these CRISPR-loaded nanoparticles into their arm veins. The liver is an easy target for lipid nanoparticles that have made their way through the circulatory system. Hepatocytes are the cells responsible for absorbing the nanoparticles in the liver. After entering the cells, the nanoparticles degrade, releasing CRISPR to eliminate the offending gene. Both conditions result from a mutation in a gene that activates a harmful protein. Intellia's CRISPR treatment for hereditary angioedema aims to limit kallikrein protein production by knocking out the KLKB1 gene in liver cells. Repeated, disabling, and even fatal swelling bouts are caused by an excess of another protein called bradykinin. An Intellia news release states that patients had 1 to 7 monthly swelling attacks before obtaining a CRISPR injection. Over 16 weeks of surveillance, the CRISPR infusion reduced seizures by 91%. Transthyretin amyloidosis is caused by mutations in the TTR gene, which leads to the production of transthyretin abnormalities in the liver. Over time, accumulating these damaged proteins causes significant problems in various tissues, including the heart, the nervous system, and the digestive system. About 250,000 to 500,000 people worldwide are living with this disease, one kind of which can cause heart failure. A patient's prognosis is often given as 2 to 6 years from diagnosis. The TTR gene is the source of the disease-causing protein, and Intellia's CRISPR treatment aims to inactivate the gene. Director of the Amaurosis Center at the Boston University School of Medicine Vaishali Sankarawala finds Intellia's reported decrease encouraging. Sankarawala believes this could have a totally revolutionary effect on the prognosis for people with this disease. There's a lot of uncertainty about whether or not these changes will stick. Leonard believes that, in some cases, CRISPR will continue to be effective even after a year. Nonetheless, the liver's cells divide and repopulate, and it is unclear if the newly formed cells will retain the genetic repair. We know that once a cell has been altered, it will remain in that state forever. That can never be undone, ever. And if there is turnover, the next obvious issue is where the new cells originate from. According to Leonard, in the liver, it originates from other hepatocytes. We believe it is permanent if introduced into the cell from which downstream cells derive their information. Due to its association with various genetic disorders, the liver has become a primary focus for researchers developing in vivo CRISPR therapeutics. Scientists from Intellia and other institutions have figured out that lipids, and other fats, can be utilized to transport CRISPR to the liver because of how easily the liver absorbs them. Two other companies, Beam Therapeutics and Verve Therapeutics, also employ lipid nanoparticles to deliver gene editing reagents to the liver. Base editing is a more precise type of CRISPR, and in July, Verve initiated a trial to treat a genetic kind of excessive cholesterol with it. However, as Leonard observes, it remains a mystery how to transport CRISPR to other cells and organs. Leonard explains, the brain and the lungs are where it's really tough to get to. It's essential to keep this in mind as you look ahead to the future. When you think about the years ahead, those are the areas where everyday lipid nanoparticle technology might not work, and you may require new systems. If scientists can ship CRISPR somewhere new, that is where it will go.